Good morning, all. We're going to be talking about new technologies today in large part. That's what the most of the morning will be spent devoted to. Then we'll be turning toward public health capacity, and then we'll be talking about technology once again and looking at the ghost technology and how it's being can be applied to investigate outbreaks and emerging trends of hepatitis C transmission, and have an example from Tennessee. It's the first pilot state health department using the GHOST web-based application. So once again, welcome to CDC. Hope everyone had a good evening. And our first speaker will be Bernie Branson, talking about current diagnostic landscape in HCV. Bernie, any comments from the moderator? Excuse me.
Great, thanks, Salim. I'd like to thank the organizers for inviting me to this meeting. Hope they won't regret it after my presentation, so fasten your seatbelts. I'm going to talk about the current diagnostic landscape, and they gave me a lot of latitude with that. I do need to start with my disclosures. I have done consulting in particular with the Gilead Sciences Focus program, which does support HIV and HCV screening, and I'll mention some of that a little bit later in the presentation. In terms of the diagnostic landscape, what I'd like to do is try to talk about what, at least from my perspective, we need with respect to HCV, what's out there, which we'll spend a lot of the morning talking about, the whiz-bang, the stuff that's sort of coming, and then listening to that, we're going to see what we would think is a really nice landscape. This is the Hilo side of the big island of Hawaii. For those of you who retire in Hawaii, they don't charge state income tax on either federal pensions or on Social Security. So if you don't want to move to Texas or to Florida, it's an alternative. And the last thing I want to talk about is, will any of this stuff become available in the United States, which for me is an important question with respect to the landscape. This is some results from the HCV focus screening program supported by Gilead. And so far, as of December 2015, they have supported tests with partners of 335,000 approximately, of which 22,000 were antibody positive, about 6.8%. They managed to do RNA tests on 77% of those people, and of the RNA tests that were done, 65% were positive. But on the right, you see what the range is of all of these things. And just for comparison, since I was with HIV, they have also supported about 2.2 million HIV antibody tests so far, of which 0.7% were positive, so that the positivity rate for HCV is 4% RNA positive out of that group compared to 0.7 for HIV. And back to the sort of topic at hand, the real issue is, I think, pretty impressive to do RNA tests in 77% of the people with antibody tests. The range is 35 to 87%. They wouldn't let me put the lowest one of the range in there, which was 12%, because of different sort of issues. And the way they accomplished this was, number one, through laboratory reflex to HCV RNA, and also by supporting patient navigators in order to get people their results. In terms of the laboratory diagnosis of HCV, this is the current algorithm, and this algorithm came out in 2013. Most people are familiar with it when the REBA was withdrawn. And for me, looking at what we need, I think, is better candidates here for the second test in the algorithm. It had been, of course, the REBA before. We went to HCV RNA as the appropriate thing, and it might be that we could use some tests down here in the corner for the people who are antibody positive and RNA negative to see whether they were truly exposed or they were false positive. But I do think the second test is important. And in terms of the second test, where we use RNA right now, I want to point out, this is from the APHL FAQs that were put out in December 2015, at which time the tests that had an indication for diagnosis in HCV RNA were all the qualitative RNA tests. And for historical reasons, I think these qualitative tests were developed primarily for blood screening. And blood screening activities were the things that have driven a lot of the regulatory requirements for this kind of testing. Hepatitis C is sort of lucky because in March of this year, the TACMAN viral load test from Roche received indication for diagnosis as well as for monitoring. Now, for both HIV and HCV, as a clinician, what most people are going to want is to know what the viral load is. And it's obviously desirable to use a test as part of your algorithm where you also want the actual results of that test. And I think that's the direction that we hope to move. And this is very recent with HCV. In terms of the experience that we've had with people going to reflex to RNA after antibody positive is a couple of things. One is there was a big discussion on ClinMicroNet about the potential for cross-contamination with these multi-platform analyzers. In particular, if you've got fixed pipette tips, like one of them does, without using replaceable ones, or the ones that do have replaceable pipette tips, they don't have any aerosol barrier. And so the question is, if you're running an immunoassay on this and you go into that tube, is it possible you're going to contaminate it when you take it over for molecular amplification testing? People are concerned about that. The other issue is the specimen handling requirements if you do reflex to RNA after antibody positive, because there are some strict requirements in terms of when the plasma has to be removed and refrigerated that are different for the different tests, 6 hours for the M2000, 24 hours for the Roche Cobas amplifier. And so what we found with most of our programs is they went to a two-tube, or tried at least for reflex, a two-tube system, where you would send in 2 tubes. They would use one to test for immunoassay, and the second one, if the immunoassay was positive, they would do the RNA on that. Now, the hospitals all found this pretty logistically challenging, and there was some real feasibility and cost-effectiveness issues, which is you're drawing an extra tube, but most people don't have more than 10% positivity, so you're throwing away 90% of those tubes. And to accession those tubes and to keep them on the side and handle them has proven very difficult for people to do that with respect to RNA. And then there are, for the people that we heard from yesterday, from Rick and Chris in the big laboratories doing 2.3 to 2.6 million tests, if they need an extra machine, they just get it. And we saw the big pictures that Belinda showed us of her things at Cleveland Clinic, but although she says it's a small hospital, it's not exactly like a lot of these places who send out their RNA tests after they get a positive antibody test, and so it's a little complicated to go through this whole strategy. In terms of HIV, just to go for a second, our last algorithm basically did also call for RNA, but it called for RNA as the third test in the algorithm. And let me tell you that nobody wants to be the third test, right? Because the first test is what everybody gets, the second test is what all the positives get, and what's left over is the third test. Well, what happened in a lot of circumstances, in fact, is that the differentiation assay would be negative and clinicians would order a viral load. Viral load does not have a diagnostic indication. Labs can't reflex to a viral load automatically unless they've done some kind of validation, because labs have to follow the package insert. So that while FDA does not prohibit off-label use, CLIA does. And so there's a different set of regulations that apply in that environment. I've always thought that maybe where we ought to be going is make the viral load the second test, because we're going to want that result essentially in 99.8% of the people who are positive on the first test. We want that result anyway. That would sort of give us useful clinical information, and then sort out the difference if we need to with the antibody test afterwards. But back to today's program, we're going to be talking about the core antigen assay and point-of-care assays for antibody, core antigen, and RNA, and then later on, integrated testing for multiple pathogens. When we look at will any of this become available in the United States, I think the landscape changes a little bit with respect to that. This is the other side of the big island, the Kona side of Hawaii, which is, a little more rocky. And when I have talked to manufacturers about this, I tend to get a response that I characterize as the 3 R's, which determines what happens. And the first R, of course, is ROW, the rest of the world. This is when they talk about the things that happen, and we'll have some discussions today from the rest of the world about that. But for example, the antigen antibody 4th-gen tests were available in Europe 10 years before they came to the US. This core antigen test that we'll hear about today was approved in 2009 in the European Union. Second R is ROI, return on investment. The situation with manufacturers is that their products need to make money. And FDA approvals are, sort of expensive, which brings us to the third R on this, which is FDA, where the R is silent in that. It's not exactly FDA. It's really regulatory burden, and it's not only FDA, but it's CDC, CLIA, FDA, who do recommendations, who do these kinds of things. The first thing I was able to publish after I left CDC and didn't have to go through clearance went into the weeds on these kinds of things with respect to the investment. But basically, if you look at an HIV diagnostic, the requirements are usually 7,000 to 9,000 specimens. Takes usually a year or two to collect them, about $1,000 per specimen just for the participants in that. The application fee for pre-market approval, HCV and HIV are both class 3 high-risk devices, and just the application fee is $250,000 compared to $5,000 for the usual FDA-cleared kinds of tests. And if you want a CLIA waiver, it adds about 1,500 subjects and about $1.5 million. In terms of lessons from HIV, how to get around this, CDC, when we wanted to try to bring a rapid test to the U.S., we did conduct a clinical trial of rapid tests and provided the data from those trials to the product sponsors. We encouraged them, and in some cases, they were able to use published data. And with HIV, at least, we did a treatment IDE for some of the initial tests where we were able to use them before they were FDA approved. When the home collection kit came out, the FDA decided to use public health impact as part of their safety and effectiveness model, and they decided also later on to look at risk-benefit models so that, for example, the requirements, which FDA states is a lower bound of the 95% confidence interval, had to be 99% for sensitivity if it was a lab test, 98% if it was a rapid test, 95% if it was a home test. And in fact, the test that was approved had a sensitivity of really around 93% with a much lower bound for that, but it was approved on the basis of its benefits for public health. So that is, I think, what we need to talk about today is how we get to the benefits. Now, Salim said the reason he invited me here is because of my telephone analogy that I'd used before with HIV, and there actually are also 4 generations of tests in HCV that have changed over time, and we have not yet come to the antigen-antibody combo test, which I think is sort of next. But that's sort of the end of the stuff I have to talk about at this point in time, and I'm looking forward to hearing about what else is out there, and then hopefully what else is going to be coming to the U.S. market. So thanks very much.
Thank you, Bernie. Questions?
God, it was 2. Well, I think the core antigen and some of the other point-of-care RNA tests, or at least near-patient RNA tests, like the GeneXpert, the Cepheid, those are all out there, ROW. You know, one of the things for me that was the biggest problem is that the best antibody test, rapid antibody test out there, the Determine test, never came to the U.S. market. You know, I mean, it was done by Abbott. They sold it to Alere. Then Abbott bought Alere back, and I think they're trying to unbuy it or something like that right now with the things that are happening. But, you know, that was a phenomenal antibody test, and it was, in fact, part of our treatment IDE initially, and Abbott withdrew and said, no, we will not bring that to the U.S. You can't use it as part of your study. And I think we get stuck with being behind very often in that regard.
Bernie, you mentioned about this issue with potential contamination or crossover by using the same tube for antibody and then followed by RNA testing. You know, we exchanged few papers.
Yeah.
So is this true for all the platforms, or some platforms do not have
I think that it's true for none of the platforms, in my opinion. You know, the study that was done with Architect, which uses the fixed pipette system and stuff like that, where they did alternating negative specimens with high positives, and they ran all those afterwards. There were about 3% of the specimens had extremely low levels of RNA, which you could tell from what the level is. The other, OrthoVitros and the Siemens test, both use pipette tips on them, and studies with those have shown no crossover contamination. So I don't think that that's a true realistic concern. However, the logistics of having a specimen in one of those, you know, when you looked at Belinda's machines, those were big machines, but they also had big paths of tubes going into these automated kind of systems, and plucking a tube out of that in order to do the second test still, I think, poses a big logistic challenge for laboratories.
Is there any downside that you would see for using the RNA quantitative for the reflex test for the HCV? Because that sounds really attractive. Is there a cost issue? Is it harder to do? Anything else?
You know, I think that the question is there a downside to using, I think there's a downside to using a qualitative test, because who cares about a qualitative test result? You know, people want a quantitative result, and the presence of RNA is by definition diagnostic of the infection that's there. And I mean, there's not a huge reason for using the quantitative test except for this determination of 8 weeks versus 12 weeks of therapy and the 6 million that sort of cutoff in that regard. But, I think that people want that result anyway, and the qualitative result just adds an extra cost in there because the next thing they're going to do in the positives is a quantitative. Thanks.
Our next speaker is Dr. George Dawson. He's the Distinguished Research Fellow at Abbott Labs in Abbott Park, Illinois. And the title of his talk is HCV Core Antigen Assay.
Oh, it started. Okay. I'm on for 10 minutes. Okay. Great to be here this morning. Actually, I have two titles at Abbott. One is manager, and the other is shown here. You can decide at the end of the talk, should I go back to the manager title? So anyway. I'm going to talk about HCV core antigen. I'm going to try to do this very quickly. I'm going to talk about the performance, test performance and claims. I'm going to review the package insert and really show that the applications shown in the field are consistent with the package insert claims that we have. So the package insert claims show here detection of acute infection in the pre-seroconversion window period, diagnosis of active infection in seropositive individuals, and there has some utility in context of direct-acting antivirals. I want to make a note here, shown at the bottom, that in the last year or so we discovered a new human blood-borne PEGI virus, HPGV2. It's the same virus discovered in Lipkin's lab, and we've done some studies with it. I'm bringing it up in context of HCV because this virus, so far, it's a Flavie-like virus. It's only found in HCV-infected people. We find that about between 1 and 2% of HCV RNA-positive people have this PEGI virus RNA. It's unknown what its pathogenicity is, but I want to call your attention, maybe something to consider as we conduct these post-forums and outcomes. So we're doing additional studies looking at the prevalence of this virus in different settings. Here's the depiction of the HCV antigen test. It's a three-step assay. The first step is a treatment step that is necessary to detect core antigen, because what's different about this test than with surface antigen or P24 for HIV is we detect antigen in seropositive people. Virtually everybody makes antibodies to the core protein. So this pre-treatment step destroys all the antibodies, breaks up the virus, destroys the lipid element that houses the core protein, and probably disrupts the core protein into bite-sized chunks. We don't know if it's monomeric proteins or monomeric pieces or dimeric, et cetera, but it does disrupt the nucleocapsid. In the first step of the assay, the pretreated sample is captured onto this solid phage, which contains 3 different monoclonal antibodies. That's a 14-minute incubation, and after that step, we add a conjugate that has 2 monoclonals that are conjugated on a complex fixture that really is a signal amplification type of device. So it really gives us the high sensitivity that we have. The test, this is an early study from 2010, just showing that the cutoff for the test is between 3 and 10 femtomoles. Is 3 femtomoles. Anything between 3 and 10 femtomoles is repeated. And if it comes again between 3 and 10, it's considered a positive. So you can see here, we detect all the genotypes. There's some slight difference in the genotype detection, but virtually we can say we detect all the genotypes. When the test first came out, it was developed in collaboration with a company in Japan and Abbott Japanese scientists. The first question, the Abbott Japan did not want to commit to the correlation of RNA with protein. They said they're 2 different things, apples and oranges. So what was done, what everybody else wanted to know when it hit the field is, what does the core protein test correspond to in terms of RNA copies? So you can see here 4 studies shown, and it looks like the 2 different methods were used. One is to take a high positive sample, do serial dilutions, test them in the core antigen test and in the RNA test, and come up with a correlation of the 3 femtomole. What does that correlate in terms of viral load of international units per mil? So you can see with the dilution of high titer between 500 and 3467 IU per mil. A second way to look at the correlation between RNA and core antigen is to test a population of samples that are RNA positive and to graph out the RNA on one axis and the core antigen on another, and look at the line that transects the 3 femtomole cutoff. And that correlates to different studies here, but you can see there's a trend here. It's between 15 and 3,000 IU per mil is where the cutoff is. If you're below 1,500, you're probably not going to detect very many of those. You'll detect some, but not too many. So our package insert states that we have a chemiluminescent microparticle assay for quantitative determination of HCV antigen in the human serum or plasma. And it's an aid in the diagnosis of suspected HCV infection. And as I mentioned, there's 3 uses: detection of acute infection, detection of HCV antigen in seropositive individuals, which allows you to distinguish active infection from resolved infection, and third, as a guide to antiviral therapy. And so everything here I tried to annotate with papers that are out there, so I want to be, upfront. So here's a depiction of the natural history of HCV infection. Right after infection, there's an eclipse phase where you can find either RNA or HCV core antigen. Shortly thereafter, within a few days, you can detect HCV RNA. And within a day afterwards, we detect HCV antigen. We've documented this in different studies. And then there's a window period, we call it the pre-seroconversion window period, of between 6 and 10 weeks, wherein RNA or antigen is detected, but antibodies are not. Then antibody comes up. And if you didn't do that disruption step, core antigen detection disappears. Core antigen would go negative, but because of the disruption step, we can detect core antigen. Here's studies that were published as part of our package launch when we launched this. I'm going to pull you to the lower panel here, and what's shown is seroconversion panels that start with an RNA negative result, and then the person becomes RNA positive, then antibody positive. And what we wanted to look at how well core antigen did, how well it matched with RNA, and in this study, the difference between RNA and core antigen is shown here as 0. So the first bleed that was RNA positive was also core antigen positive. And you can see here the days earlier detection versus antibody was about 35.8 days. So about 5 weeks earlier detection with an antigen test than an antibody test. Here's a study that was recently published, and I know Claudia from FIND is a co-author of this, and there may be another, the main author may be in the audience, I'm not sure. Yes, okay, thanks very much. It was a great paper, and I've got a couple slides here. It's really a very comprehensive look and an unbiased look at the HCV antigen test. And here's the claim showing acute detection, that is samples that are antibody negative and RNA positive. And you can see the overall sensitivity was between 92 and 98%. I'm sorry, was 92%. For specificity was 98%. I'll call attention to that Reyes-Mendez paper here, where it's 0. There were only 3 samples, and they were all less than 1,000 IU per mil, so no surprise, we wouldn't detect those. Here's another recent study, Lepers et al. From Transfusion, comparing the sensitivity, analytical sensitivity of the Ultrio HCV RNA test with 2 combo tests and the HCV antigen test. And you can see in terms of copies per mil, the Ultrio detects 3.5 copies per mil. The combo test, 3.3 million for MONALEESA, 3.4 million for Murex. The HCV antigen test, 2,700. This is in beautiful correlation with what I showed you earlier. There's a constant theme here, whether you're looking at populations of samples or analytical sensitivity. Here's another study showing we're more than 1,000-fold more sensitive than the combination antigen-antibody tests. Here's what we're suggesting as a potential algorithm. Several different papers have come out and shown this from, and I can show you the different papers, but I think 4 different sources. We kind of adapted the CDC algorithm here, the icons, and showed HCV antibody negative result, you stop. Antibody positive, we're saying go on to a core antigen test. If it's positive, you're reactive, your current infection, go on to treatment. If you're non-reactive, you can reflex to an HCV RNA test. So that's what we're proposing, and some customers do that today. So here's another depiction again from that paper, Freeman et al. 2016. It's a great paper, and we show here the sensitivity and specificity, again, about 92%, 0.5 across the studies, 97.8% specificity, and you can see most of them are in the 90s. This one paper is an outlier. I don't really know why that is in a Chinese journal. So, but that's the big picture. constant theme. We don't detect everything. The samples we miss, often less than 3,000 IU per mil. So current guidelines for monitoring therapy rely heavily on RNA testing. We know the landscape is changing. We know that there may no longer be a need to monitor with a viral load test, to have a quantitative viral load at each step of the way. And in fact, before the direct-acting antivirals, there were more test points than is now currently being proposed. We're looking at a scenario where you do an antibody test and either viral load or core antigen for confirmation of active infection, and then a baseline type of test, whether genotypes will be needed anymore when these antivirals come out that will work regardless of what genotype it is. And so you do a baseline, and then you would look at perhaps at 4 weeks post-treatment an adherence test, and then at 12 or 24 weeks after treatment an HCV antigen test or viral load. So that's what we're looking at, and we've done a comparison study here wherein we've looked at the concordance between HCV antigen and RNA testing on an actual trial with the AbbVie SAPPHIRE study. And we can see here the overall concordance is very high, greater than 99%. In the pre-treatment, I kind of flagged here the statements, the samples that were different. There was one sample that was missed that had more than 1,000 IU per mil, missed by the core antigen test. Here, 2 samples were detected by RNA and missed by core antigen, and there were low titers. Here, 6 were antigen positive and RNA negative. These eventually resolved, but a lot of times at the end of treatment, you do see some residual core antigen. Here's the correlation coefficient between RNA and HCV antigen, showed at 0.74. And just to summarize, again, you know, we can detect exposure pre-seroconversion period. We can distinguish active from resolved infection in most cases. We missed a low viral titer. And we've shown some data that with the emergence of direct-acting antivirals, the core antigen test could be used. Okay, that concludes my presentation. Any questions?
The talk is open for questions now.
Yes.
Yeah, George, I have a couple of questions. One is, how long does the test take, and does it run on the same platform as the antibody test?
Yes, it does. It runs on the i2000, Architect i2000. It's a typical test. It's an 18-minute first step, 4-minute second step, but there's a pre-treatment step of 7 minutes, so it's about 30 minutes.
And the second thing, we heard a lot yesterday about the idea of using basically something like an antigen test just to do one-step screening for somebody, and you say here, at least possibly, it would pick up people in the window period, but the algorithm you proposed was using it as the second test in the algorithm after antibody. Do you want to address the differences?
Well, it's like, would you do RNA testing on everybody coming in the door? Would you do core antigen testing on everybody coming in the door that you suspect of viral hepatitis? It would be a very expensive endeavor. So an antibody test is less expensive. It's kind of a first screen. We think that a good combo test that had it lower than 3.3 million would be a good alternative to have a good combo test out there that would detect most of the early seroconversion window period. Then you could reflex to an antigen or RNA test. But it'd be very expensive for everybody coming in the door to run HCV antigen, especially because it's an expensive test to make. And it was actually proposed to do that in Japan because the prevalence was so high to say, they had this program called Health Check, where they're proposing, instead of doing antibody, then Reba, then RNA, to do the core antigen test up front on everybody. Just one test. If you're positive, you go on for evaluation of liver function. If you're negative, you're free. Anything else?
George, you mentioned the limit of detection in terms of HCV RNA, it's approximately 2,500 international units. Is this true for all genotypes? Because there are some studies which show that it may be much higher for genotype 3.
There is a subpopulation of individuals infected with genotype 3 that have amino acid polymorphisms that probably 2 to 4% of people. We know we've looked at databases from thousands of isolates, and we can see that there are amino acid differences in some areas that can reduce our detection, such that instead of having 2,500 IU as the cutoff, it's probably more like 30,000 or 50,000 for a subpopulation of HCV-infected people. It's probably 4 or 5% of the total genotype 3 samples. And in fact, we revised our package insert to call attention to that in some cases, there's a subpopulation of HCV-infected people with genotype 3 that may be difficult to detect, that have medium or low titers. Yes?
I know most people don't know how much anything costs here apparently, but I was wondering, just relatively speaking, you said the antibody is much cheaper, but is it 4 times cheaper? Or just to get an idea, maybe at what prevalence, starting with core antigen screening.
I don't do
would make
that type
sense?
of math, no. I don't like to answer those type of questions, probably two to fourfold more expensive, depending, there's different prices and different, things, so I don't like to answer those, I get myself in trouble. Yeah, right.
I have to say, though, it does seem like, George, in high prevalent settings, first-line core antigen test could be more seriously considered. It might well be cost-effective.
It would be good, and you have to define, what's, you have to do the math.
Exactly.
The cost of antibody plus RNA versus RNA alone, or antigen alone, and what the prevalence was. If prevalence is 50%, you'd probably do best by doing a core antigen test, but if it's 6%, I don't know. So you have to do that.
Right, and the drop-off and the cascade along the way as you lose people because of the availability of a PCR test versus having the core antigen right
Yeah,
there.
I mean, we are very much aware and would love to be able to come to a situation in U.S. where we could come with the test, but that ROI type of thing is, return on, it's very expensive to come to the U.S., and it's not a huge market. It's a subpopulation of the antibody positives would be tested with this.
Thank you. Any further questions we can keep for the roundtable at the end? Thanks, Dr. Dawson.
Our next presenter is Dr. Michael Reed. He is the Senior Vice President R&D and Chief Science Officer at OraSure Technologies. Mike?
Hey, thanks, Salim, and thanks very much for inviting me here to speak. I think OraSure is a familiar name to quite a few people here, maybe not so myself. I've been with the company for about 4 months, so very happy to be able to be part of this. So I'm going to talk today about the benefits of rapid testing in at-risk populations, and to talk a lot about the test that OraSure has developed to support this market in both the global and public health and whole country elimination campaigns. So everyone's aware here that identifying the HCV positive individuals is a critical first step to eliminating HCV. You've got to know where the infected people are before you can treat. And for at-risk populations in particular who are resistant to going into doctors and healthcare and being tested, an important ability to test outside of that environment. So the OraSure product, very, very simple to use, with many sample types. So in the US, the product's approved for fingerstick and venous whole blood, and in Europe approved and globally approved for all 4 sample types. So in the case of oral fluid, it's a simple swab around the mouth, place the device into a developer vial, and in 20 minutes you'll get your result. With blood products, there's a loop that pulls out 5 microliters of sample, mix in the developer solution, put a fresh device into the developer, and you'll get your result in the same time, 20 minutes. So a positive, a non-reactive result will generate a single line in the control zone. So this tells you that sample's been added to the device and it's flowed properly through the device and is present. And then a reactive device or a reactive sample will see 2 lines appear, one of the control zone, again, to confirm that the test performed properly, and then at the test line you'll see a line which indicates the presence of HCV antibody. So the operating principle is really, it's quite simple in concept, quite difficult in practice to develop a really robust test in this platform. So the sample is loaded on the pad. You see the antibodies there. If HCV antibodies are present, they'll start migrating through the device from right to left here. And there are, I guess, three key zones in the device. So first, you have a conjugate pad, which contains colloidal gold conjugated to protein A. And this protein A will then pull out or bind to any antibodies in the sample. As it migrates up the device, it'll then hit the test line, which has a permanently bound HCV antigen. So we're using peptides and antigens from core NS3 and NS4 proteins. So any HCV antibodies bound to the conjugate in that situation will then react with the HCV antigens and create a stripe. And then the control line, again, is anti-human antibody will bind to any conjugate complexed with an antibody. So at the end of the day, if you have a positive sample, you'll have a stripe appearing at the test line from the concentration of the gold conjugate antigen antibody complex, and your control line will react as well. So the performance of the product is really quite remarkable and comparable to lab-based EIA tests. And so the data shown here is to support CE marking of the product, and you can see in the different sample types here, very high sensitivity and specificity. I think one thing to note in the oral fluid, there were 8 of the 12 false negative results in the oral fluid sample were also PCR negative, indicating a resolving infection when that was tested. Something which is also a feature of the test is its ability to detect seroconversion before the lab-based tests. And so you can see in the bottom panel here an average of 0.6 days before the EIA reference, but significantly more panels were detected first. So this has been repeated in many external independent studies. In 2013, the Walter Reed Institute evaluated 29 different rapid tests, 5 of them in performance studies, and in all cases, the sensitivity, specificity, and seroconversion sensitivity were superior. And so some of the data from that study you see aligns very well with the clinical data that we collected to support CE marking, with sensitivity and specificity here very high and equivalent. You see the confidence intervals and the observed values. And then again, with the seroconversion, 3-day detecting samples 3 days earlier than the lab-based test and well before the other rapid tests. In 2015, there was a meta-analysis of 30 different studies, a nice compilation of all the studies that were out there. And again, AuraQuick outperformed all the comparative tests in these areas. Also pointed out, and we had some discussion yesterday about the impact of a HIV co-infection. I will point out that in the CE data set that we collected, there were 230 HIV-HCV co-infections and 830 HIV patients in that data set. So we weren't seeing an effect in that data set. Maybe in some global settings, there's some other factors we need to consider. So the test is obviously very capable and a great format for the public health setting and for use in high risk and more resource poor environments. And so just a quick overview of where the test is being used. It's been deployed. Over 3 million tests have been deployed in over 45 countries worldwide. There's a large multinational NGO, and we heard yesterday from Médecins Sans Frontières that they're using our test very successfully for their initiatives. They're very proud to be a part of their efforts. We've also recently signed an agreement with a foreign country to supply millions of these tests through 2017 for elimination initiatives that they're running. In addition, domestically, you can see that there's been a really widespread state health department adoption over the last 4 years, with many jurisdictions adopting this test as part of their outreach testing programs, as well as other non-government testing locations where at-risk individuals are present. So, and we're also supporting and part of a number of multi-city initiatives and supporting many efforts in this testing community. So, in summary, so we're testing testing for and identifying HCV-positive individuals really critical in at-risk populations for elimination campaigns. We have OraSure has developed a point-of-care test shown in many global studies to be best in class, and is providing a much-needed solution for the humanitarian and public health needs for HCV elimination campaigns. Thanks. I'll take any questions.
So, Mike, let me start. Yesterday it was discussed, this issue of false positivity in African populations. I'm just curious, have you tested any samples from any of the populations there and found that you have similar false positivity with your test?
So we haven't seen and I think that there was a suggestion of false positive results because the confirmatory testing rates were identifying confirming less positives than you would expect from the antibody results. So I think we haven't seen certainly not a significant impact, no, and we're not aware of the data that we saw yesterday. So we hadn't seen this.
Same question about, as there's some studies that have shown that the HIV co-infected and even pregnant women, their false negative results with most of the screening assays. So have you tested similar populations with your?
Not a large number. I think the HIV, HCV co-infection situation, I know in the standard, the at-risk populations we tested in our clinical studies were certainly not showing any impact. I think the impact of co-infection and immune-compromised situations, people under ART therapy, it's been known to impact sensitivity, but not aware of a large data set. So we have talked with FDA about the studies needed to support that claim. I think at the present time, the available market and the additional volume that would come from oral fluid testing does not really support the cost of the submissions.
Antigen test?
So yeah, so we have evaluated the antigen an antigen test in here, and I think given the challenges that Abbott has had in developing this test with an instrument with really rigorous sample preparation to expose the antigen, it's kind of unrealistic to think that a rapid test could solve the problem that they're having. So we've evaluated it, not working on a development program at the moment. But yeah, good question. Certainly would be a nice program to figure out how to do. Yeah.
I had just a general statement about there's been talk of false positives in African setting and other tropical settings, and I just wondered, just as a potential to look into, is the impact of heat being very, very hot in these places, that if a sample sits at 37 centigrade or higher, this could perturb the ability of the, could impact specificity. So you might look at it, in those people doing studies in those type of remote places, is draw the sample, do the test immediately, let it sit as if, you weren't going to test it for a couple of days, test it again, and see if it goes from negative to positive. It may be something like that, because we've seen it in our own labs when we import samples in from Cameroon and different places like that we see false positives, especially in an indirect assay where, if the antibodies are warmed or heated, they can stick to the solid phase, and your conjugate is anti-human IgG or IgM is going to find that. So just a general, comment, you might look into that to see if you test it immediately and then let the sample sit for a couple of days, would you see any difference?
Brian, you have a question? No? So since we have time for one more question here, have you looked at TBS elevates for the detection of antibodies using your device?
Not that I'm aware of.
Okay. Okay. Thank you, Mike.
Thanks.
The next speaker is Dr. Hong Ki, she's president of Q Labs in San Diego, California. And the topic of her talk is point of care, HCV, core antigen test development.
Good morning. I'm going to show you some of the recent results that we've been working on developing of the point-of-care rapid test for HCV core antigen. This work is supported by the SBIR contract from the CDC. There are a lot of challenges for HCV core antigen detection by immunoassays. I have listed several of them here. Sensitivity is usually associated with the quality of the antibody and the method of the immunoassay. So I'm not going to spend time on discussing the sensitivity issue here. Rather, I will focus on the genotype diversity, the properties of the HCV core antigen, and the host antibody. HCV virus is very diversified. There are 7 genotypes and even more subtypes. To develop immunoassays, the challenge is how can we use a small set of antibodies to cover majority, if not all, of the genotypes? Fortunately, although the HCV virus, the polyprotein itself is highly diversified, the HCV core antigen is more conserved. We analyzed over 3,700 HCV sequences of the 1A and 1B from HCV database. We found that in over 85% of the core antigen, there are only 5 amino acids that are not conserved. If we align the consensus 1A and 1B core antigen to the other core antigen subtypes of other genotypes, we also found multiple stretches of amino acids. Multiple stretches of amino acids that are identical throughout many of the genotypes. If we develop antibodies against these identical stretches of amino acid, then it would be possible with combinations of antibodies to multiple epitopes, it would be possible to cover all HCV genotypes. Let's move on to the HCV core antigen. This protein is not an easy protein to work with. It aggregates and precipitates in solution, and many of the recombinant proteins are unstable. This caused a lot of difficulties in developing immunoassays. Further, the host antibody made the immunoassays more complicated. The core antigen is very immunodominant. It is very immunogenic. So actually in the patients, when they are producing the anti-HCV antibody, there is a good part of the antibody is anti-core antigen. So in the patient sample, the core antigen is mostly in the immunocomplex. So with the problem of the core antigen itself and also the host antibody, how can we develop an immunoassay to effectively detect the core antigen? Currently, the solution is, as Dr. Dawson just pointed out, you have to treat the sample, dissociate the core antigen from its bound antibody, and then wash away the antibody. Then the core antigen is further detected by its specific antibody. So this process is very complicated, and it's apparently not adaptable to rapid tests for point-of-care detections. We come up with the idea of developing immunoassays under denaturing conditions. If this is possible, then we could dramatically simplify the immunoassay protocols, and it would be possible to make rapid tests for the core antigen. However, for making an immunoassay to work under denaturing condition, we would need antibodies that also are fully functional under denaturing conditions. Fortunately, this is exactly what Coolabs specialize. We work on single-domain antibodies. Camelid family of animals, they produce a special class of single-domain antibodies. These antibodies were first discovered by Dr. Hammer over 25 years ago. The heavy-chain single-domain antibodies, comparing to the conventional antibody, it does not have a light chain. It does not have a light chain, and it also lacks the CH1 domain in the heavy chain. Further, the variable domain of the single heavy chain antibody is fully functional in terms of binding its antigen. And this part, the variable domain of the heavy chain antibody, we call it VHH. This VHH section is only 15 kilodaltons. The smaller size of this antibody gives it a lot of advantages. First, it is very cost-effective in production. It could be produced in many available expression systems. And also, because it is naturally produced by the animal, it has mostly high affinity. Many of them has nano to picomolar range of affinity. And also, because of the smaller size, the VHH single-domain antibodies has very unique flexible paratope structure. They bind molecular clefts and cavities. Also, they would bind epitopes that are different from those of conventional antibodies. Most importantly and more related to the work we are doing here, these antibodies are highly stable even at temperatures of 60 to 80 degrees, or in the presence of denaturants, such as 2 to 3 molar guanidine hydrochloride. And even after being denatured, they can easily refold to fully functional antibodies. Here, I'm showing you a simple experiment demonstrating the VHH single-domain antibodies being used in immunoassays under denaturing conditions. We made a lateral flow assay test strips using a single-domain antibody and also its antigen. We use a goat anti-rabbit antibody as a control for the conventional antibody. Here in this result here, when we applied PBS or 1 molar guanidine hydrochloride, you see both the VHH antibody and goat anti-rabbit antibody are able to form immunocomplex forming the 2 lines. But when the guanidine hydrochloride is increased to 2 or 3 molar in the test strip 3 and 4, only the VHH antibody is able to bind onto its antigen, forming 1 single band here. The goat anti-rabbit antibody does not work anymore. This experiment indicated, first, the VHH antibody can be used in constructing rapid tests such as in lateral flow immunoassays. And also, these immunoassays can be used in denaturing conditions. How do we make single domain antibodies? I'll just quickly zoom through this process. We immunize llamas with selected antigen. By the time the llama produced a good antibody, we would purify the PBMC, which contains the B cell, and then purify the RNA and synthesize cDNA from these B cells. Then we use specific primers to PCR amplify the heavy chain coding region. Because the VHH antibody does not have the CH1 domain, the size is actually smaller than the conventional heavy chain antibody. So we could select the VHH coding DNA and clone it into a phage display vector to make a VHH antibody library. After a few rounds of feed panning based on antigen binding, we could identify positive clones, which contains the specific antibody. Now that we have the plans and strategies in place, we synthesize the multiple groups of peptides and use them to immunize llamas. Here I'm showing you several llamas that we immunized. And the first llama apparently produced more antibodies on the peptide 8 and 9, more on 9. And the second llama mostly produced antibodies on peptide 1 and 2. And the third llama produced antibodies across the board throughout the core antigen. We also used expressed the full-length core antigen to immunize a rabbit. This rabbit produced antibodies mainly to the peptide 5. We then used these llamas and purified the PBMC cells and constructed the VHH phage display library. Here is showing you the QC data for each of the library. Currently, we are working on isolating the specific antibodies from these libraries. So I just showed you the work we did on developing rapid tests for HCV core antigen, and we are working on developing multiple antibodies for different epitopes to cover the genotype diversity, and we are using llama single-domain antibodies so that they can be used directly in immunoassays under denaturing conditions. With that, I would like to acknowledge the staff at the CDC for their scientific input and also the funding support, and AbCor Inc., which is our exclusive partner for llama immunization services, and all the staff at CoolLabs who had worked very hard and made significant contributions to this project. Thank you all.
Thank you. We have time for a couple of questions. I had one question. When you immunize the llamas, is that with denatured antigen, or is that with the native structure of the antigen?
Okay. We immunize the llamas with the synthetic peptides and also the full-length antigen, which is expressed but purified under denaturing conditions. So it's all fully denatured.
Okay, thank you.
A quick question for the upfront processing. Did you have to do any sort of wash steps or any heating or anything during the denaturation?
Like for the experiment I showed you, we didn't do anything. We just applied the 3 molar guanidine hydrochloride directly onto the test strip. And that's what we are hoping for. We'll just treat the sample and then apply it directly to the immunoassays.
Yeah.
sorry, Gilberto Bajon, Division of Viral Hepatitis. A very quick question. I'm not that familiar with this kind of antibodies, but considering that you don't have the light change, how that affect the specificity of the antibody, considering that the FAB is much shorter than antibodies in other species?
Yes, the heavy chain antibody, the variable domain is actually larger, especially the CDR3 region, which is thought it is extended to compensate a lot of function of the light chain. In fact, in the conventional antibodies, majority of the specificity is provided by the heavy chain part of the antibody. And the light chain is mostly for structural purposes, and it hides the hydrophobic part of the molecule so that it's more stable. And that is also thought to be compensated by the extra long CDR3 region.
One quick question.
We have time for one quick question.
Hey, Hong, oh, sorry. Go ahead, Mustafa.
Good morning and great work. And I'm sorry I missed the first part of this talk and very good work. But I'm just wondering if you already started using normal human serum, could be normal or could be infected, because what I can see right now, antibodies are produced in lambs or llamas and recombinant antigens, but haven't you started using human samples in this work or not yet? That's all.
So you are asking if we have tested human samples?
Or during your experimental phase, the kind of phase you are in right now, you started using serum or human samples yet?
I'm sorry, I couldn't.
I'm sorry. Okay. Have you started using human samples? Could be positive or could be negative for HCV or not yet?
Not yet. Yeah, this work is all preliminary, and we are using the artificial samples.
Artificial samples. Okay. Thus, we need. I'm just. I'm sorry. Curious about the interaction that you will get from real human sample will be the same as you are doing right now, or you expect it to be different. You know what I mean? Because whenever we are doing such a kind of experimental work using commercial antibodies or generated antibodies or antigens at the first trials of our work, when we start moving to the next level of this work and start using human samples, real human samples, we got different feedbacks, we got different results.
Yes,
you know what I mean?
exactly. Yeah.
Thank you.
One thing we did consider is that the real sample would be much more complicated than the artificial sample, which mostly are made with PBS. And so in the lab right now, we are testing using animal serums spiked with the core antigen, just to see how, in the presence of a lot of antibodies and other proteins, how that would affect the denaturing conditions. That's where we are now.
Great. Thank you, Dr. He.
We'll have time later on.
Thank you.
So firstly, I'd like to thank the organisers specifically for allowing us to share our work with you today. It's a great honour. And I'll be speaking today about our assessment of the point-of-care RNA tests as a strategy to enhance diagnosis and linkage to care, particularly in drug and alcohol settings. So this is not the audience to convince with regards to how important screening and diagnosis is to improve this, the HCV care cascade. And we've already heard this morning about core antigen work, rapid antibody tests, and this presentation is about point-of-care RNAs. So the other thing that we've also discussed is how important alternative sample types are to particular patient populations. We've heard about oral fluids for the rapid antibody, but also dry blood spots is an area of interest personally for us. And today I'll be talking about capillary blood tests. So the data is clear, and not only is HCV treatment of people who inject drugs effective, it's also critical if we want to achieve global elimination. So in addition to removing the restrictions around treatment access and stigma, we need to increase community awareness amongst this population and improve screening, diagnosis and treatment access, particularly in drug and alcohol clinics. So we also discussed yesterday how important it is in terms of the structure for diagnostic testing, whether you use centralised or decentralised settings, depending on the patient preference and the capabilities of the country. So it's clear that for people who inject drugs that we do need to shift towards a more decentralised setting to reach this population. So I don't need to go to detail about the HCV, the Expert HCV RNA cartridge. It's essentially a cartridge that contains all of the reagents you need for amplification or extraction, amplification and quantification of HCV RNA. And this lends itself to a couple of points with regards to its capabilities for point-of-care testing, and that is it's self-contained and single-use, so you limit that cross-contamination issue we've been discussing. It also is automated, so it reduces hands-on time considerably and minimises the training required. It's very simple to use. The time to result for the new cartridge coming up is 60 minutes. The one I'll be talking to today, the plasma cartridge, is 108 minutes. And an aspect of the platform and the cartridge system that I think is often overlooked is its ability to, it's random access, so you can put samples on at any time and use any of the cartridges available for HIV testing or any other infectious disease. And it comes in multiple configurations, from the sort of coffee espresso machine through to the AT Infinity platform. But the overarching question, of course, the big question is whether it'll be affordable to those who need it. So we decided to incorporate the RNA test into our Live Alive health promotion campaign, which is also assessing FibroScan as an intervention to try and improve HCV assessment and treatment in drug and alcohol settings. So the first stage of this program was to develop messages regarding liver health with focus groups with the affected community. And then we used these campaign tools in a campaign days, which was essentially we go into the clinic with a team, and that team consisted of peer support workers, clinicians, nurses, and our research team. Sounds like a lot of people, but actually wasn't that many. And we'd set up these booths, and we engaged the people at the front with coffee, which is very important in Australia, and food. And then once they were enrolled, we then, they went through and did a questionnaire on a tablet, a self-assessment, pre-assessment. We did a finger prick assay, collected dry blood spots and venipuncture for the point-of-care RNA into a minivet. FibroScan, and these results were then discussed with the nurse, a standard of care test, and also another research sample for plasma collection, and a follow-up questionnaire. So I'll just be referring to the plasma and the finger prick tests today. So the methods are fairly straightforward in that we used venous whole blood standard methodology, collecting it and storing it 4 degrees, and processing it and testing it the next day on the Cepheid machine. And we also collected finger prick, which is a standard Lancet collection, into the little minivet, which is 100 microlitres, loaded that straight onto the plasma cartridge, diluted it up to 1 ml with a buffer, and then ran that on the day of the campaign. And we compared both of these results to Abbott real-time as the gold standard in plasma. So for participant recruitment, the first lot of campaigns, which included a supervised injecting facility in Sydney, we only collected dry blood spots. So for the RNA point-of-care testing, I'll be referring to these results for 182 participants, and they're collected from homelessness services, 3 OSD clinics and a youth service along the east coast of Australia. So the results here are looking at the expert viral load results in plasma and finger prick compared to Abbott using the Bland-Altman plots. And so the Bland-Altman plots, you can see they measure the difference between the expert minus the Abbott for plasma or venipuncture. And you can see we've already got an outlier here, which was positive by finger prick and plasma on the expert, but negative by the Abbott. So if you remove that outlier and expand the axes here, you can see that the data is fairly well, it's quite tight amongst the 95% confidence limits. I think it's a range of half a log plus or minus there, roughly. The diversity, you can see the dispersion is fairly even along the concentrations, but we do lack the number of low viral loads. And the mean, although I don't have it plotted on here, is not far, the bias is not far from 0. So with sensitivity and specificity, if you use detectable as the cutoff, you can see here that the expert in plasma, compared to the Abbott in plasma, we have one discrepant result, a sensitivity of 100% and specificity of 99.1. And in the finger prick, we get 4 discrepant results with a sensitivity of 95.9 and specificity of 98.2. But this result here is the discordant result, which was described on the previous slide, the outlier. So if you remove that, then you do still get the 3 discrepant results. The sensitivity and specificity doesn't change much. And they are at low viral load that you can see here. If you exclude those on treatment, because a number of these people are receiving DAAs already, it's not a specified time point. So if you remove those, the sensitivity and specificity, don't actually change much. But if you then change your cutoff value to quantifiable and unquantifiable, you can see that we still get 1 discordant result. And the same with the plasma, you get the single discordant result, and the sensitivity and specificity is around 98% and 100%. And again, if you exclude those on treatment, the sensitivity and specificity doesn't change significantly. So we then were able to ask a proportion of these people, 92 people, about their acceptability of the assay. What do they actually think of it? 88% of those said that they liked their results the same day. If we asked them how acceptable it was to have a finger prick test, 88% said that it was very acceptable. Of those people who said that they prefer a finger prick, we asked them why, and they said because it's quick. Majority was at 62%, but a quarter of the people, and that was a message that I was anecdotally hearing, is that it's hard to get blood from these patients, and that is the case. And the nurses would take 2 nurses half an hour to get a sample for the research pad sample. And then we asked them which method of testing with regards to sample and time would they prefer, and 57% of those people said that they prefer the finger prick with a result in 60 minutes. So this is only like an initial preliminary data set, and obviously we don't have any data to support whether or not the point of care assay actually links more people to care and achieves more higher number of cures. So the future directions for this particular study is that we're continuing this work with the new cartridge I'll describe in a moment, with additional 300 people, and including an Aboriginal medical service in Australia. We have a similar liver life study in Bangkok planned. And I'm not sure if you're aware, in Australia we have unrestricted access, and we're doing a rapid scale-up of DAAs across Australia. And we're incorporating a simplified liver life with the RNA testing in Ethos Engage with 2,000 to 3,000 people beginning in March. So the cartridge, as we've used at the plasma, which is standardly available now, has been improved with support of FIND and Unitate by Cepheid. They've removed that dilution step that we used and reduced the time to 60 minutes. So that's the cartridge that they're going forward with, and it's currently being tested in the field. So the study has limitations in that we've got potential selection bias, obviously, because these participants are already engaged in care, and a majority of those are people who inject drugs. Limited sample size, obviously, it's a small study, and particularly we have a few samples at the low viral load, which is clinically relevant for a number of reasons. And access to venipuncture was a significant issue among this population. And time to result also limited, but we had to get a number of extra machines to resolve that. So finally, in summary, the finger prick provided a simplified practical workflow in the field. It had pretty good sensitivity and specificity, and simplified sample collection and shorter time to result is preferred by the participants and also the staff. And the new finger prick cartridge is being tested in the field at the moment with a shorter time to result. So the Liver Life campaign has been an extraordinarily fun effort, a big effort by a number of people. We even tried it in a van, it didn't quite work. The communication cut out every now and then, but these are the people involved across the board, and I particularly like to thank Jason Grevely, my colleague, who leads this project with me, Yasmin Mowat, the clinical trial coordinator, and Francois Lamerie, who did all the laboratory work. So happy to take questions now. Thank you very much.
Thank you, Claudia.
Gilberto.
So very quick question. I'm more familiar with the cartridge for TB, and I know that the company is increasing the number of targets, like reporting all the mutations for drug resistance and all that. For HCV, the cartridge, there are plans to include genotyping for the virus?
No, because as you've sort of discussed, I think thinking further ahead that the genotyping may actually disappear with the pan-genotypic, so I don't think it's in there. I don't understand that there's plans to include genotyping in this assay.
I'm off again, sorry. So you talked about the field testing being started now, so can you give a little more about that, about what settings you're testing it in and how you're doing
Yeah,
all
so
that kind of
there were similar settings. So we've been involved in at the alpha trials, which is similar setting to the drug and alcohol settings. So there's other laboratories around the world involved, and I can't speak for them, I'm not sure. But then there'll be, I understand, moving to beta testing shortly, so that's all I know. Yeah. But similar settings as far as field testing in those environments.
Bernie?
The Gates Foundation actually just announced yesterday that they're sponsoring field tests of this in a sort of a large way, but no one has talked yet about how much it costs.
Oh, so I don't know how it will pitch. I heard yesterday it was $16. I mean, I'm an academic. I have no idea about the commercial aspects of this, but I heard yesterday $16 was is that right, from MSF and FIND? So I'm guessing that's equivalent, but again, that's still out of range for a lot of people. So hopefully by the sheer volume, if they get their market share by volume, they'll be able to compromise perhaps with regards to... Do you have a comment on that, Claudia?
Yeah, so the $16 is a fine negotiated price. So it's for low and middle income countries. It will be obviously more in the US. Yes, there's a volume target-based pricing. And it's going down to, I think, 13 is the lowest. They won't go any further unless something else happens.
So I think in developed countries, they're still wanting, they claim will be more affordable, so they'll want to pitch their market and get the market by affordability and beat the price of the standard tests.
Can you talk a little about the logistics of actually using the machine one cartridge at a time,
Yeah.
and how many machines you needed to run the study, and what you think you'd need in
For our
routine
particular
practice?
study, it was really important that we recruited people quickly. So we actually tandemed a number of the assays. So when we had the 108-minute cartridge, we used 3 machines together because in the initial, so for this population, they're busy, they're not very patient, they want to leave. But the study itself took an hour, so we had to get the results done in an hour. So for that reason, for them to circulate through all the booths, so we wanted to have that limited to an hour. So then when we had shorter time to result, we wanted to keep the recruitment going on the day because there's a lot of momentum and energy. People want to get in and out. So we actually tandemed about 6 machines, but that's only it's proof of concept. So, but in terms of using the machine, it's honestly a beautiful machine to work with. It's very simple. And the only time we, with regards to utility, had a problem, and it was our fault, we pushed the boundaries a little bit, which we used the camper van, and then at one point someone stepped in and went, and they all started dying, so we went, okay, that doesn't work. But we probably didn't set the camper van up very well. So.
Tanya, how long does the sample remain stable in the mini-vet, in the capillary, before it has to be transferred?
So the mini-vets, to start off, we were using mini-vets that weren't coated with EDTA, and so we were very cognizant, we were worried about clotting. That might affect the machine, so we had to put that on straight away. With the mini-vet, they're actually now coated with EDTA, which you can keep it for longer. The cartridge itself, once you open it, has to be used within 30 minutes. We were putting the mini-vet on straight away, but it would be a very good question to ask as to how long it could sit there for 30 minutes, an hour, or whatever it is before it's put on the machine. So I don't have the data for that, but it's a good question.
And in your comparison studies with Abbott platform, was the sample input volume the same for Abbott test as well as your test? You said the concordance was...
No, so these results were adjusted, the dilution factors were adjusted. So 100 microliters was adjusted to 1 mil, and we used those factors into account when we compared the results. But the expert cartridge uses 1.1 mil, and I think the Abbott used 500 microliters for their particular assay. So we adjusted the calculations accordingly.
Okay. Thank you very much.
Thank you.
So we're going to make a slight adjustment to the program. We're going to take the currently scheduled break and put that after the roundtable. We'll go forward with the next speaker. and she is Dr. Christiane Honish. She's a director of microbiology. Director of Microbiology Markets at Illumina in San Diego, California, and her talk is titled Integrated Testing for Multiple Pathogens, Virome Analysis.
Thank you very much for the introduction, and thanks for the organizers for inviting me. I'm actually stepping in for a colleague of mine, but I'm happy to always come here because I worked with a hepatitis group before in a different role. So I'm going to talk about not a diagnostic tool at this point in time. I'm talking about next generation sequencing and looking into virome analysis with it. And I think it's going to give an introduction to our next session, which we are going to have here. So next generation sequencing basically is a high throughput platform and sequences very deep into viruses and resolves quasi-species, and that's what we are talking about here for HCV. There are multiple different library preparation protocols out there, which were originally and heavily at Illumina, where I'm working at, developed for the human genome, for human genome sequences. But we are translating those into the analysis of viruses, into the analysis of bacteria. So going through that list here, whole genome sequencing is what we now using also for bacteria. The FDA food outbreak monitoring has adopted that to actually track our food. To sequence all these bacteria, Salmonella, which can, Listeria, which can cause food outbreaks, and have built up a really nice tracking system throughout the US and globally to actually monitor and catch outbreaks even before they happen. Then in humans, there's whole exome sequencing. Then we will hear more, I think, later on about amplicon sequencing, where you use targeted regions and you sequence very, very deep. Transcriptome, RNA, so you actually look at transcripts, and that takes us into viruses already, RNA viruses, and exon capture in human sequencing. And really the idea for a capture approach came out of this approach, where what I'm going to talk about is capturing viruses in a mixture. So there's one form of sequencing where you can sequence everything in a sample, but you get a lot of human background. And for samples like HCV, you really want to resolve the population structure of the virus. And that's where it's very, very helpful to enrich these samples. And that's what I'm going to go into. You actually capture a whole gamut of viruses with a lot of oligos, and then you sequence to decipher what you captured. And when you compare the two methods of sequencing really everything in depth, you get about 1% of viral population versus human background, whereas when you enrich, you can get up to 80% of virus, or even over 80% of virus, depending on the viral titer, of course. So if we go through this process, the process is called RNA access workflow, where you start with the RNA, which gets fragmented, you produce cDNA, and then you prepare a second strand, and the double-stranded cDNA, you add adapters, which are amplified by PCR. And then in the next step, there are actually two hybridizations. So you do a hybridization with these specific oligos, and the pool of oligos can be up to 50,000 oligos in a pool. And I'm going to show you on an example how we'll tile across the whole viral genome or multiple viral genomes to capture as much in the sample as possible. And these oligonucleotides are captured with magnetic beads, so that you enrich for the virus. This hybridization process is done usually twice, and then you amplify again and sequence on a platform of choice, all the way from the MiSeq to the HiSeq. So I would like to give you an example which is published, and I'm going to show you at the end one example which is really data which I got from Lilia Ganova-Reva yesterday on HCV, where we are just stepping into that. So I don't have much on HCV, but I would like to give you an example a different virus. So for Ebola, we developed a panel where you can see, as I told you before, we are not only targeting one virus, we actually looked at multiple viruses which are covering the Ebola virus, but then closely related viruses as well. And we are tiling across all these viral species to build a pool of oligonucleotides for the capture. So this is how it looks like for one example. Here you have the Zaire Ebola virus genome, and in this case, we put 230 oligonucleotide fragments, which are about 80 base pairs long, tiling across the whole virus. And we do that for all the viruses I've shown you before, or even for a bigger pool, to capture the variability of the viral population. Then we go through the workflow, and I would like to give you one example. This is actually my colleague Gary Schroth, who couldn't be here today, where this was used for a very interesting story in Ebola. So at the end of 2014, the Ebola outbreak was going down, and there were actually in 2015, there were only 2 cases of really prevalent viral transmission, which came up again. And one of these examples I would like to show you here. So just a few points about Ebola. So it's detectable in the bloodstream, usually during and only during acute illness by quantitative PCR, but it can persist in immunoprivileged sites for longer periods of time. In breast milk, for example, vaginal secretions, eye fluids, and in semen. And the WHO had set a certain time frame upon which an individual, a patient, would no longer be infectious. And in this story, it's actually shown that, unfortunately, it persisted longer in semen in a patient. So the case and the sad story around this is the following: that on March 20th, a 44-year-old woman was confirmed with Ebola virus by qPCR, and the patient died 7 days later. So there was no immediate contact because the whole large outbreak was declared to have calmed down. So in this case, she was infected by unprotected sex with a survivor on March 7th. So the whole story started with the onset of the survivor on September 9th in 2014. His blood, and he actually, as I said, he survived, so his blood was tested negative on September 29th, and then again confirmed on October 3rd. So he was surviving, and then he had the intercourse with his girlfriend on March 7th, and as I said, the patient sadly died 7 days later. So how did we try to determine if the survivor was infected, actually infecting his girlfriend by sexual intercourse? So for whole blood, as I said, we did sequencing of the patient. We got whole genome sequences with good coverage. We sequenced also the survivor's brother and the survivor's wife. The survivor semen was attempted to be sequenced with a normal sequencing library prep, but we didn't get any data. So there were no sequence data obtained for that, but then we used the process which I showed you before, the enrichment process with this panel, and we were actually able to decipher this transmission and the epidemiology of it. So the results were, we have the survivor up here, and there was just one mutation difference between the survivor and the patient. The survivor's brother was formerly infected and had passed away from the same strain, whereas the survivor's wife lived somewhere else and had was infected by a different strain. So the genomes from patient and survivor differed by only one nucleotide, as I said. And 3 substitutions absent from all other West African sequences was actually the survivor's wife. So this is just a representation that, in a way, we got very sensitive, but we also got very lucky. And I wanted to make this point. So this wasn't, these single nucleotide polymorphisms weren't found by a high coverage. So parts of it, we only got 1 genome, we got 5 genomes. And here you have the survivor, which basically has all these mutations, and the patient, which is overlapping basically with the survivor except for 1 mutation, but only with a low coverage, which we would have never detected with normal library prep. And with that, I would like to close with just one HCV slide. So we are doing these panels against other viruses, against other quasi-species. We've developed a panel just for research purposes on respiratory viruses. I'm reaching out to you. We are open to collaborate on different research questions. We are working with Yuri's team and Lilia on hepatitis C one. And this is just the very first design, brute force designing based on quasi-species sequences. And as you can tell, just on one example of a genotype 2B, capturing, we get a pretty even coverage. We now have to go back and see where don't we get coverage, where do we need to do more work on adding oligonucleotides. But this is just really getting sequences from Lilia, designing it, and running it. It's a very promising tool for the future to decipher genotypes and mixed genotypes in sequences. And with that, I would like to thank you for the attention, and I'm open for questions if we have time.
So you used 230 oligonucleotides for Ebola. So for a 10 kb HCV genome and you want to do a pan-genotypic assay, do you have the capacity to add that many oligos?
Yes.
I mean, what is the maximum? Is it thousands, 10,000? Or?
Yes, it's thousands. It's about 50,000. That's what he said.
Okay.
So at some point you might run into oligos overlapping, but we designed them in 20 base pair concepts. I'm actually very interested in testing this, and I'm looking at Claudia here for TB, to pull out TB genomes from sputum, but we haven't tried that yet. But basically, we can add enough oligos to pull a lot. And the hybridization sensitivity is not that high, so we don't tune it exactly to a certain temperature. We tolerate about 10% mismatch. So it's basically like a sponge where you absorb everything which binds to that sponge, but it's not 100% specific. That's what we get through the sequencing later on.
Okay, and I know it's a little off topic, but can you do this for DNA when you're looking at integrated HIV proviruses? I mean, I would assume you can do that.
That's what I would love to try. If you do a fragmentation, I think you could do that, yes. Thank you. It would be interesting to do it on the boundaries if you know the integration, right? Yeah. And differentiate, yes.
I had one question, but could the other speakers please come up to the podium? In terms of HCV, if you have mixed infections, do you think this type of technology can distinguish mixed infections for HCV potentially?
Yeah, that's the hope. These are the initial examples where Lilia just used pure genotypes, but that's the hope. And then looking at recombinations within the viruses potentially. Yeah. So and for the pan-viral panel, you would put different viruses in there. And we are planning on putting, here there are different genotypes in there, it's just shown in one.
Right. What sort of ratios of viruses do you think you could, if you have a large concentration of one virus versus a minor, say, HCV genotype?
My hope is that we pull everything in, and that's why we have to tune this in equal amounts. The sequencing will give you the resolution of the mixture later on. So that's why you don't have to be 100% specific. On the, you might want to maintain the ratios to a good extent to detect the minor ones, that's true, but the actual resolution of that mixture is going to come from the sequencing.
Okay, thank you.
So we have about 15 minutes for this roundtable discussion, so if you have any questions. So let me start with the two presentations on the core antigen, George and Hong Ki. There was this recent publication in hepatology from Professor Kyun, who they described that this step of dissociation actually decreases the sensitivity of the assay. I just wonder if you have any comments, thoughts on that assay. George?
I'm not familiar with the conditions used. Can you tell me any more about what was used?
The methodology was not described in detail, but what was highlighted, I think it was also last year in the ASLD meeting, this paper was presented, and what was highlighted is that this step of dissociation brings down the sensitivity, and I do not understand how, so I thought if you have any thoughts.
Yeah, I don't know. You know, it's very difficult because we don't know how many core molecules are associated with a virion, probably several hundred. And so, you could decrease an antibody-antigen reaction, but you could, in breaking this complex macromolecule up into bite-sized pieces, provide more targets for the capture and detection. So I think it's a difficult question to answer. You know, we don't know how many core proteins are associated with each RNA. So you may get more benefit from dissociate, even though you bring the ability of antibody to find antigen down. You revealed so many more targets with the dissociation that it overrides that.
Alberto?
A quick question for Christian. Good to see you, Christian. Back again. Over here. So I gather that you guys are still developing the whole thing, right? And so I can see that, adding the primers and also the hybridization and all that stuff, that seems to me at least that it's going to increase the price, right? So the pre-processing, I'm not talking about the sequencing per se. So just bulk numbers, I know it might be difficult for you to get an estimation, but price, cost. Because, you know what, I'm asking you because other approaches like SureSelect or HaloPlex that were targeting sequencing, they are, I mean, for whatever I can remember when we were working with 454, they were sort of expensive.
Yeah, I actually don't have a price for you right now, but we are working on producing oligos, and we have the whole array business. So we are trying to get the oligo cost to a decent amount, but it's not going to be a test which it's not a fast test, so it's not a diagnostic test, and B, it's going to be hopefully inexpensive. We get it down to the array front, but it's not going to be a $15 test for the diagnostic purpose. This is more a research tool.
Thank you. My name is JJ Jackson. I have two questions, one for Dr. Appellate, and the other one is for Dr. Houchet. Let me start with her first. I think your presentation was very nice, and I like that. The test, the model the sequence you're using is testing people who are drugs addicts and on alcohol, and that is a sample you are using right now. I don't know. Will you have similar results? Were you to test people that are now within that bracket? And my question is coming from. I'm from Florida, and what we do is that from our little county, we look at a specific community that is high rates with all of these different problems, and then we carry all of these STDs, HIV, hepatitis C tests on them because of the situation. We feel that that community, because of unemployment, poverty, disease, and stuff, they should have these things. And our result is showing that our negative rating from that population is very low, as compared to the environment, the community that we consider to be a high-class community that that have a higher level of education, nutrition is good, everything is good, and we have a huge level of positive in those areas. So my question is that if you are only looking at substance abuse people right now, is it possible that the positive you're getting right now could also equip to the areas that you're not testing if you were to carry that test on that stuff?
So I guess the question relates to the networks that are involved in the transmission for the hepatitis C amongst those communities. So in these studies that we were performing, each site had different prevalence. So the homelessness setting, the prevalence was 25%. In the drug and alcohol settings, it was 50 or 60%. And on the first day of our first drug and alcohol setting, an OST clinic, it was 80%. And we're going, oh no, like this is not good. But I think it was biased representation on that day. If we then move, I would expect that the transmission networks, the social networks amongst different populations are very different. And we have evidence for that because there are other marginalised groups that we work with, HIV positive MSM, and we also work with prisoners as well. And the networks are extremely different, therefore the prevalence is different. I can only speak to, for example, our experience with the HIV positive MSM cohort group, who they do have a reasonably high positivity rate, but they are also in Australia highly educated, engaged in care, they're all working. But there's a high positivity rate in that population as well. So yes, the prevalence will vary amongst different populations. I can't sort of explain why you're seeing the difference, a lower prevalence than you'd expect in the drug and alcohol setting compared to the higher educated population. I can't necessarily speak to that, but the networks will be very different. In the injecting drug network that we see in Australia, it's very localised. You inject with your partner, you inject with someone you've been buddies with for years. Whereas in the HIV positive network, you do all these sequencing transmission studies, phylogenetic work. The networks are much broader. It's the different circumstances under which the networks are transmitted. So you have a higher number of people that you're connected to, but it's a small population. I'm not sure that answers your question at all.
Bernie, I had a question about, did you review any HIV-HCV combo test and what's known about their performance? So I think Myra had an HIV-HCV combo test, so anything about their performance?
A couple of places have looked at the combo test, and to be honest with you, it really depends on what the format is, so that in the one you're talking about, the MedMyra, which was an immunoconcentration or flow-through test, it lends itself better. The lateral flow tests, I mean, there's not something now, the dual path platform, which is better for multiple antigens, but everything until now has shown that the combination tests have slightly lower sensitivity than the individual tests do.
Thank you.
I think this question is mostly for Dr. Cui because you talked about it, but maybe Drs. Reed and Dawson have a thought too. For the guanidine, that's a hazardous material, right? So how do you capture it on the strip and how do you dispose of it if you're going to apply this in a point-of-care setting?
That's a good question. Right now, we are mostly testing it in the lab, and we haven't really gone that far yet. And we are actually testing multiple denaturant and all different harsh conditions that you can imagine. So in the end, we probably will have to take that into consideration, like how do we put it into the test so that it's more user-friendly and it's more environmentally friendly. It's a long way to go.
So I think that's it's a very real problem, and I think there's also the risk that once you get into human samples where you're trying to dissociate the converted patients, the antigen in the seroconverted patients, that you're going to need some heating and other pre-treatment. And every step that you add to the upfront processing takes it further away from point of care, right? So that's a real challenge, I think, that the lateral flow or the rapid test technology platforms are going to face with this.
So, George, and it's also for Mike, yesterday, Ray Kim mentioned that about 22% of the patients, kidney disease-related patients, that they were seronegative but were RNA positive. I was just wondering, is there any studies planned to look for core antigen as well as to use your OraSure rapid anti-HCV test to see if that test can detect antibodies in them? Since you showed that compared to the screening assays, the OraSure test detects antibodies like 35 days earlier. So I was just curious whether...
No, the antigen test.
Antigen, sorry.
Detects 35 days earlier than conventional antibody testing.
So have you looked at that population, this like the seronegative, they remain seronegative?
We've
for a
looked at
long time?
seroconversion panels that are available from Seracare, serial plasma donors. They collect plasma, every few days, and then at some point it's found to be RNA positive, so you have all the backlog you can test. And we've looked at those. Like I said, we matched RNA testing on many of the panels. We published all this. We've also looked at what's called NAT yield samples. The American Red Cross does blood screening, as you know, for the last 15 years, 14 years, something like that, for HCV. We've tested 240 RNA NAT yield samples. Remember, there's only about 4 NAT yield samples, 4 RNA positive, antibody negative per million, 4 per million United States. So we tested 240 of those, and we detect about 92%, I think, something like that. And then we've looked, we haven't yet looked in kind of just the diagnostic setting in a high prevalence area, but that's where I think the value of a good combination test will come in, is to be able to detect people who are walking away, that are RNA positive, walking away with a negative antibody test result, that are actually carrying the virus and probably highly infectious. But a good combo test, I think, will be of value to use in the diagnostic setting.
Thank you.
The Hep C summit, but you spoke on Ebola, and I'm from that region. I'm originally from Liberia, and so I'm very much interested. The survivor, what were you investigating? Were you investigating him as a carrier, or because his wife died from the virus, and he had a girlfriend, and the girlfriend was also also a patient. However, he was still negative. So, what were you investigating at that time? Him as a carrier, or I don't know the science on that, please.
So he was cured, and then his wife was infected and died, and his girlfriend got infected after he was declared cured. So the WHO has set a certain time frame where patients are considered to be cured, and I should have made that point clearer. Actually, the time when he infected his girlfriend was past that time when he was declared cured. So the transmission happened through semen, and that's one of the body fluid where the virus can be, well, if you can detect the virus, where you can detect the virus longer. So the CT value for the semen sample was actually 38. And you can't, with normal library prep, you can't, in normal sequencing, without this enrichment trick, you wouldn't see anything. So we were lucky to see a couple of genomes, and in some cases only one not even full genome, where we were happy to get these individual SNPs and mutations, which allowed us to build the phylogenetic tree and actually trace back that contact to a sexual contact, which was important because it's the first case where it's showing that the time frame which the WHO said it needs to be looked, the patients need to be looked at longer, especially in these body fluids. So I hope that helps to clarify it.
Tanya?
I just had a question, actually, if that's okay, in relation to that. So I understand you had paired samples, so semen, but no paired blood at that time. But at diagnosis, you had access to the original positive blood sample. So understanding that semen is an area that is protected as such, are you able to sort of make any, it's more of a research question rather than a diagnostic question with regards to the evolutionary pressure of that compartmentalization that may occur in semen? How different was the semen sample to the original blood sample when he was originally diagnosed?
So they were the same for the regions which we were able to sequence.
So you had limited data. If you had
so
more
we
data,
had limited
yeah.
data, and that's why I showed the table to set it in perspective. Yeah. We really got only single breeds, but you didn't get any information with any other technology. So I wouldn't infer a lot more than that
it would be
from
wonderful
this
to
data.
see the evolutionary pressure in the different compartments.
And actually our collaborator states that in the patient, that out of this finding there should be far more systematic studies following.
Definitely, that'd be great.
To really look at how long is the transmission time really.
Mm-hmm.
But luckily the outbreak died, but it's something to look at if this is ever coming up again, as exactly like you were pointing out.
So similar studies in HCV with regards to semen and transfer in looking at specifically HIV positive gay guys, but I was wondering about the pressure in that compartment.
Mike?
Yeah, so this is, well, I guess I wasn't expecting to be talking about Ebola today, so it's a little bit off topic, but we've received a grant from BARDA to develop an Ebola antigen test. We have emergency use authorization for a rapid test that has also has an intended use for oral fluid in cadavers, and we're currently working on seminal fluid as well as an indication.
All right, great.
So it's that's just starting, but we're in active development on that rapid test program. The product is in use today, although the outbreaks are pretty much dissipated, and but it is being used in surveillance programs in Africa today to monitor for outbreaks.
Yeah, we've deployed our first system there to actually and trained individuals on site to actually do epidemiology in Africa on site. So,
Bernie?
Panel's asking each other questions, I guess. George, you mentioned about using a combo test in order to pick up people who were earlier, but if it requires dissociation for the antibody, how do you make a combo test?
You don't need, for the combo test, what you do is, you put a reagent in that reveals the core antigen, but it does not destroy antibodies. So we've done this, we've published on it in the past, and that's what Murex and Bio-Rad do now. We had a prototype test several years ago, and so we know how to do that. There are reagents you use. If you don't use a specific type of reagent, you won't see the core antigen in the pre-cell conversion window period. But you can reveal that antigen with the addition of certain chemicals. And so what you do is you detect antigen in the first part, then your antigen detection goes down, and your antibody comes in. So you are able to bridge that gap between probably when you get RNA titers of 2,500 IU per mil through chronic infection.
So to clarify, you would be able to get essentially two results from the one sample? Or you
We
just
don't
get
have
a
that
reading saying it's
capability.
fine?
Some manufacturers do, which is really cool.
Yeah.
But we get a single readout, so you won't be able to distinguish which it is.
Which is what you need to know, right, if
Yeah,
they've
so
resolved
you're just
or
going to
not.
know that person has been exposed to HCV. They either have RNA or antibody.
So if you had the two readouts, that would be perfect, wouldn't it? You could tell whether they've had it, but they've resolved.
It would be, yeah.
Like in the rapid test, there are quite a lot of products that you could actually make multiple test strips on one test. So you could apply the sample to the first test, the second test, the third test, and you get the readout simultaneously. And then you would be able to tell, say, for the case of HCV, you would have a readout for the antibody and another readout for the core antigen.
You could do that, I think
ideal.
Determine does that, don't they, Bernie? Yeah. The antigen test's not too sensitive, though.
Right.
Yeah, it can be done, you're right. You can get antigen, antibody on a strip test and distinguish.
It's from, for OraSure, this is from one of my summer students who used the OraSure rapid test in the lab. So one question was, do you plan to develop any sort of smartphone app that you can take a picture of the results and it converts into positive, negative results? You don't have to wait. There is no inter-operator variability. And the second question she asked, now that we're all, worrying about the environment, trying to go eco-friendly, why is there so much plastic in this device? Can you cut down on the plastic on all sides and make it more sleeker and thinner? Since we have the CEO in the audience, so I thought.
Great question. So we were at a meeting a couple weeks ago, next generation diagnostics meeting, and the need for connectivity solutions and ability to transmit data from rapid tests is certainly being acknowledged as a pretty urgent need, and with different I guess, end goals in mind. So in developing markets, more to monitor outbreaks and location of devices and tests and so on. And in the US and Europe, more around transmission of data to EMTs, EMRs, and so on. So yes, we're certainly evaluating some devices, and we'll be very, very keen to associate a reader with our test. As far as the plastic, so we would love to take more plastic out of our device, but there's certainly some constraints around that. There's a ton of work and regulatory implication around that. But it's certainly something that we're looking at, and you know, cost reduction is top of mind for everything we do. We have a version of our HIV tests in use in studies in Africa for self-testing programs, and have sort of slimmed down the packaging into a pouch format and so on. So we're able to provide Africa, and which is looking like it's going to be a really a nice product for the needs of the various 90-90-90 initiatives going on in various African countries.
But the device still has plastic?
Yes, it does. The device is still...
So the box is gone.
The box is gone, but the device still has the plastic, yeah. So we were actually just talking about this the other day, but yeah, it's a real challenge to make any change to the device, as you're well aware here.
So if there are no other questions, one for Han. Is llama the only animal that produces these single domain antibodies?
The camelid family all produce the single domain antibodies, like camels, llamas, alpacas. It's just llama is easier to live in California, I guess.
Than a camel.
Yeah, and also the single domain antibody has been found in sharks, and I guess llama is even more easier than sharks. Although we have SeaWorld there, but yeah, we have a farm. AppCore has a big farm there with over 300 llamas now.
Okay, so with that, I'd like to thank the panel for a very interesting session.